some of the work that we've done looking at copper mine adaptations in the yellow monkey flower. And so the study of species that grow on mine tailings is actually a really classic system that's been used to study rapid local adaptation for a number of decades now. It's been a really good system to use because often we know when these mines arose, so we know exactly when the environment's changed. And additionally, selection is often very strong in these mine habitats. And there's a very abrupt change between the healthy normal soil and these contaminated soils. And so one observation that's been made repeatedly is that very few species grow in these heavy metal contaminated soils, despite the diversity we might see immediately adjacent. And so this has led many people to ask, why are some species able to adapt while others are not? And so we have attempted to address this question using one species in particular, and that is Mimulus cutatus, or the yellow monkey flower. And this has also been studied. This species that grows throughout Western North America. In particular, there are populations that grow on old mines and mine tailings throughout California. And these populations have been studied for a number of decades as well. And so there's really a rich body of research behind them already. So what do we already know about copper tolerance in this species? Well, we know that it's characterized as root growth. So Allen and Shepard in the 70s collected plants from mine and off-mine populations in the field. They grew them in hydroponic copper solutions and found that tolerant plants had normal root growth, whereas non-tolerant plants seem to have inhibited or restricted root growth. And they also found that this trait seems to be fixed in mine populations. Following their work, McNair found that this trait seems to segregate as a single dominant locus. And more recently, Kevin Wright has found that this locus, which we call TAL1, increases survival by 30% in the field. So this has a huge effect on survival and on fitness in the field. So despite all that we know already, one thing that we don't know is what genes underlie this trait. And that's certainly not from lack of trying, and so Kevin Wright, in addition to his field work, tried mapping this locus, this TAL1 locus, using 4,000 intragress lines. And he found that this region maps to linkage group 9, which is like chromosome 9 in our system. However, it falls into this region that's a paracentromeric region, and so there are a lot of repeats, and thus this region is unassembled in a reference genome. So trying to map for these genes without a reference assembly has proven to be really hard and almost impossible. And so today I'll tell you a little bit about what we've done to try to get around this issue and find candidate genes that are in this TAL1 region. So if we can't do traditional mapping, what if we compare tolerant plant and non-tolerant plant and see if there are any genes that are differentially expressed? Well, we can do that using an RNA-seq approach. And so going into this project, we actually have a really great resource in these introgression lines that were used for mapping. They're made by back-crossing repeatedly a tolerant individual into a non-tolerant perennial background. And for this experiment and the others, we used one line in particular that's depicted here, and this has the smallest introgress region that contains the TAL1 region and our closest genetic marker. And this will become useful when we do our analyses because instead of having to look throughout the whole genome, we can focus on this region that we know contains the TAL1 locus. So using this line, we can self it to generate alternative homozygotes. We can then grow these alternative homozygotes in a nutrient solution or a solution that contains copper. Let them grow roots, and then collect the roots and pool them by genotype and by treatment. And then we can compare these pools using RNA sequencing, and this is done in collaboration with the JGI and Hudson Alpha. And so when we do this, we first see that most of the genes that we find are differentiated between genotypes. And in fact, there are very, very few genes that are differentiated between treatments. So what this means is that the genes that we're finding here are constitutively expressed regardless of treatment, and the biggest differences are between genotypes, so really, for now, that's all we need to focus on is that comparison. And so throughout the genome, we find that there are about 200 differentially expressed genes. But if we focus in on that TAL1 region like we can do, we find that there are about 37 genes. And only 3 of them seem to have interesting heavy metal related functions. The one with the highest fold change is this multi-copper oxidase. And so this has an 8 to 12 fold higher expression in tolerant lines than non-tolerant lines, and it's constitutively expressed regardless of treatment. And I've done some RT-qPCR, and I've confirmed these results as well. So now that we know, or we see that this gene is differentially expressed, what's causing that? Is there a copy number variation, or is it a regulatory difference? Well, we can start to address this question, and we can do that by doing some PacBio sequencing. And so this allows us to get really long reads, up to 50 kb. And our goal in doing some PacBio sequencing was to try to sequence through that paracentromeric region that contains TAL1 to try to get an assembly. And that way we don't need to try to align those really short Illumina reads that often fails when there are so many repeats. And so we just got this data back, but the results seem to show that in fact, we do find 3 to 4 copies of this gene in the tolerant genotype and only 1 in the non-tolerant genotype. And so this is pretty convincing evidence that there is in fact a copy number variation here. Because this ratio is not really the same as that 8 to 12 fold change we just saw for the RNA-seq, we do think that there is also regulatory changes going on, though we still have to work on that. We don't know if it's a cis or trans change or what they might be. But this is really exciting because a lot of recent research in metal tolerance that's come out has found that both copy number differences and regulatory changes have been involved in tolerance. Okay, so now we see that in these 2 genotypes of this introgression line that was grown in hydroponics, there's this differentiation in the MCO multi-copper oxidase gene. But if this gene is really involved in adaptation to copper mines, we might want to see or look at natural populations to see if this gene is being selected for. And so we can do that using a population sequencing approach. And this is using data that was previously collected by Kevin Wright. And so this data set contains SNP data for 2 mine populations and 2 paired off-mine populations. And so what we can do if we want to find a signal of selection here is look across the genome and see if there's any region or SNP that has a big allele frequency difference between the mine and off-mine populations. And so if we look across the whole genome, we do in fact see that there are many regions where there is this differentiation, this divergence in allele frequency. And one interesting thing that you can see is that here this is the first mine-off-mine comparison, and this is the second mine-off-mine comparison. And these peaks of divergence are in exactly the same places. And I can talk about what that means later if you'd like. And so among these peaks, we do in fact find this multi-copper oxidase gene. And so there's a big difference in allele frequency in both mine populations versus their paired off-mine populations. And so we might think that there is selection going on in the mine populations. And so hopefully I've shown you that copper tolerance in the TAL1 region is driven at least in part by this gene, multi-copper oxidase. It has a higher expression and a higher copy number in tolerant lines versus non-tolerant lines, and it seems to be selected for in mine populations. But now the question really is, what is this doing? How is it conferring tolerance? And so we know that this is a copper-containing protein. It's most similar to this Arabidopsis protein, LPR1, and what that protein does, it's responsible for recruiting callose to the roots, and that's a polysaccharide involved in cell wall synthesis. So what MCO might be doing is it might be promoting this callose deposition, which then blocks up the channels that go between cells and stops the transport of copper that way. Alternatively, this class of genes is also known to be involved in lignin biosynthesis and degradation, and increased lignin has been found in other metal hyperaccumulators, shown here in this class B species versus the Ceratopsis species. I've started doing some initial imaging, looking for increased callose or increased lignin, and I haven't yet found any. So a third and maybe simplest hypothesis is that this gene is simply acting as a chelator, binding free copper in the cell and preventing it from being harmful to other cell functions. And so my next immediate goals are to see if this is true, to see if there's sequestration, and if so, where in the cell, if it's in the cell wall or vacuoles. And I think I should be able to do this with some ionomics and maybe some more imaging just to confirm these. So with that, I'd just like to thank the people that contributed to this project.